We're going to go ahead and try to wrap up chapter 17 in this video. I think we can probably get it done. We might push that 20-minute time limit a little bit, but I really think we can do it. So let's talk about changing gene frequencies and changing uh, gene flow in a population. The first topic we get is genetic drift. Genetic drift is a random change in an allele's frequency over time. And it's kind of weird. We call it drift because it's not really driven by anything necessarily. This is just chance alone. You might get allele frequencies to change just a little bit more um, in a population, especially a small population. Your book has a pretty cool graph on page 280, and it's basically in a small population, genetic drift is can be significant, whereas in a large population, genetic drift is really not a big deal. So like humans, we're over 7 billion strong. Genetic drift is probably not going to affect our population too severely. Whereas if you have a population of animals, let's say, and there's only 15 or 20, genetic drift can be a real problem. Excuse me. So a fixed allele is a condition when all your individuals are homozygous for an allele. And so if everybody has the same exact allele, then that allele is fixed because it can't leave and no one else can replace it. It is literally the only allele available. So this would be, let's say, all your flowers are big B, and there's no heterozygotes and there's no recessives. They're all big B, big B. That allele is fixed in that population of flowers. And so that frequency can't change because that's the only allele everybody has, unless you get a mutation, which could change the allele, or some other individual with a different allele enters that breeding population. That could change that allele from being fixed as well. Oh, and here's the graph I was telling you about. Perfect. So each line is a different population, and the population was kept at 10, and so they measured the allele B plus, and so in one of them the allele B plus drops down and is completely gone. In some, the allele B plus goes to 100% and becomes fixed, and in others it increases just kind of slowly to a little higher percentage, maybe 70–80% of the population. Now these guys were kept at 100 individuals, and this is the same graph. It's the same generations, it's the same percentages, but you see how smooth this is. When you have 100 individuals, the genetic drift is not this drastic, chaotic, could be fixed, could be extinct kind of nonsense. It's actually a steady, smooth line. If you do a best fit line, oddly enough, both of these graphs have about the same slope on a best fit, but the changes are certainly less drastic in a large population. So let's talk bottleneck. If you watched my little video about my opinion on how evolution fits into a Christian worldview and whatnot, I mentioned that Noah's Ark would have put a lot of stress on the populations because it would give us a really drastic bottleneck. And I didn't explain what it meant. So here you have it. A bottleneck is a drastic reduction in the population size. And what this does is it reduces genetic diversity. Northern elephant seals in the late 1890s, they're pretty easy to hunt because they generally find the same beaches to rest on year after year. So when the hunter finds that beach, then they can kill all the seals. So we were able to drop their population significantly, and the hunting actually reduced the population to only 20 surviving individuals. And then we said, okay, no more hunting, or they're going to be extinct. And so those 20 individuals had a bunch of babies, and the population has recovered, but we took what was a very genetically diverse population of hundreds and thousands of animals, and we dropped it to 20. And so now all the babies that are born from just those 20 have a very tiny piece of the genetic material that's available compared to the whole population. And so a lot of alleles that were present in the population are gone now because there's such a small population. So a small population is not always a good thing. It really can be a troublesome thing. So speaking of small populations, you'll get this as in the founder effect as well. Founder effect is when a small group establishes a new population. Most books will give an example of somebody arriving on an island, like bird or lizard, with a hurricane, clings to driftwood and is washed to new island, right? And then they start a small population on the island. They're the founders of their population in a place where nobody's been before is kind of the idea. Excuse me. So if the founding group is not representative in terms of allele frequency, which if you're from a large population, I'm not sure how you could be, neither will the new population be representative, which means the new population genetic material is going to depend on who shows up and makes the babies in this new area. No duh. Um, we we see the founder effect goes hand in hand with inbreeding, because if you're the only two birds who land on this island thanks to the hurricane, there's not a lot of options with who your kids are going to breed with. You're going to be pretty inbred. And that's breeding between close relatives, which I'm sure you know. And what you end up getting is people who have high rates of being homozygous for recessive alleles. And a lot of times this is bad. It's bad for genetic diversity, it's bad for diseases, that kind of stuff. Because there's so many humans, you don't really see this in humans a lot, except for with the Amish. Because the Amish came to America as a small group. They have this really cool rule where they don't let any outsiders into the colony. And so you have the founder effect where these small groups of people come to America, and because you don't ever let new people into your colony, it doesn't take too long until you look around your little Amish town and realize that everybody's everybody's cousin, and you end up inbreeding because there are no options except for the outside. You don't want to go there. And so what you get is this really cool deal where the old order Amish in Lancaster County, Pennsylvania, have these super high rates of this disease called Ellis-Van Creveld syndrome. And it's a genetic disorder that gives them their kiddos dwarfism, polydactyly, 6 fingers on each hand, not that big of a deal. Some heart defects, a few other symptoms, and they've actually been able to track that back to two Amish who came to America in the 1700s. And because they didn't bring any new crop into the dating scene in the Amish world, eventually you get pretty high rates of a disease that probably shouldn't be persisting, but it's certainly persisting there because we have put together this magical pair called the founder effect and inbreeding in humans. So it does exist with us, even now. So kind of neat. Gene flow. How do we move genes around? Individuals in natural populations, they tend to mate most frequently within their population. So basically, pick any population, a population of turkeys in the Black Hills or deer or people or whatever. But nearby populations may sometimes interbreed with them if they can kind of get in the same location. And so that would be an example of gene flow. Gene flow is a movement of alleles between two different populations. And this can be a good thing. It can result in change, or it can stabilize a change because it's bringing in more diversity to a population that didn't have those genes in that population. And so certainly wind transfers pollen from pine trees. Animals transfer seeds, bury nuts for winter, and then lose them. The tree might not be able to drop its seed much more than 10 or 15 feet, but certainly a chipmunk or a squirrel can carry that seed 100 yards and bury it and then lose it, and that's a way of moving that tree into an area where it might not have been otherwise. So that's even something simple like that can be gene flow, putting a tree somewhere new. It has the potential to breed with somebody new. It might take a few years, but it has that potential. That's gene flow. So let's talk about how we can make some new species, because evolution is neat and all, but really that idea of making a new kind of animal or a new species is really very difficult. And there are a few ways we think it can happen. So speciation is the process in which we get a new species. Now, if you know much about the definition of species, it's actually kind of a difficult thing to define. It occurs when populations do not interbreed. We're pretty much all happy with that. If you can successfully interbreed, you're the same species. If you can't interbreed anymore, then you're not the same species. If you stop interbreeding, then you're going to become more genetically different with time, which makes sense. And over time, we become so different that we call them different species. So populations that might have already diverged may come together and interbreed again, and then we take all of that carefully crafted species sort of vocab and we pretty much just toss it because horse is a different species than a donkey, but they can make a mule, so I don't know what's going on anymore. It just doesn't make sense. It's not clean cut kind of lines. So one way we can do this is we can reproductively isolate them. Reproductive isolation is just what it sounds like. We're going to split these two populations up so they can't have sex anymore. And if you get that job done, then they can't make any more offspring, so they have to become a different species over time. There's lots of things that can keep two individuals or populations from successfully breeding. It could be temporal isolation. That's one example of this. That's different timing. If one flower opens up 2 weeks after a different flower closes, those guys just physically can't make a baby flower together. They can't make a seed. Some bee is not going to carry pollen on his legs for 2 weeks. This is not going to work that way. So if the timing doesn't line up, then you're not going to be able to successfully interbreed. You could certainly compare this to maybe rut in deer. If the rut for whitetail is different than a rut for muley, then their odds of interbreeding is going to be super small. How about ecological isolation? Maybe we can split them up due to adaptations. They just live in a little bit different environment. Somebody can survive, maybe a bacterium can survive an environment that's just a little bit darker than a bacterium that likes a little bit lighter. Bacterium's a really bad example because they just cut themselves in half. How about fish? We'll go with that. Fish that can live in an area that's darker, fish that likes to live in an area that's a little bit lighter. They might not get together to breed just because they're just hanging out. One's 100 feet deep, the other one's 4 feet deep. They might not get together to breed. They're just in different environments. Even though they're technically in the same lake, they just don't overlap a lot. So that over time could actually give you different species. Behavioral isolation is another good one. If your behavior differences could prevent mating, a lot of times the female's choosing the male, and if you're a bird and you build a nest that you think is awesome, and this girl bird from a different species looks at your nest and that's not what she's looking for, no babies for you. That's behavioral. Mechanical isolation is the simplest. Probably the size or the shape of reproductive parts prevents mating. So just anatomically, they do not fit well together. It can work for bugs. It can work for all sorts of different flowers, certainly plants, that kind of stuff. That's mechanical isolation. And then some can even become isolated because their gametes are just incompatible. Some flowers can tell this if they're getting pollen from themselves by accident, they won't fertilize themselves. Some flowers, they want pollen from a genetically different plant because that's the whole benefit of sexual reproduction is that diversity. And so if you get gametes with molecular incompatibilities and they can survive, most of the time they can't, but if they can survive, they can make offspring that are hybrids, and generally you're going to see some reduced fitness. The mule is the mix between the horse and the donkey. Most of your mules are going to be sterile. If you're lucky enough to get one to breed, it's going to be a less healthy, earlier dying animal than you had before. And if you're lucky enough to somehow get that one to breed, then that thing you're making three cycles down the road for the great grandkid is going to die like pretty much right away. And they just get worse and worse and worse. These hybrids aren't in very good shape. The liger's the same way. If we really try hard, we can get a lion and a tiger to breed, and they make a liger, and the thing's not healthy, and it's got all sorts of problems. They don't live that long. We can make them because they look cool, but really they're not very successful in becoming a new species. And so here's some different ways that species can form. Individuals can reproduce at different times. They can reproduce in different places. They can ignore or don't get the cues for sex because their behaviors are different, or they can be physically incompatible. If mating occurs, and a lot of times you don't get fertilization, they're different species. If you mate and zygotes form, the hybrid embryos might die really early, or the individuals might die before they can reproduce. Then you're not the same species. If the hybrid individuals or their offspring don't make functional gametes, like they're sterile, well, then you're probably a different species because that kid you just had is the end of the road. If you somehow, by just great luck, fit all of these things in and none of those things stop you, you can have successful interbreeding. It is very rare, but it happens. It happens with plants. It can happen, you know. So here you have the same thing I just showed you, except for it's a little bit larger and easier to read. So let's talk about some other ways that things can become two different species without just talking just specifically breeding. We can just physically separate these guys. Physical separation between populations. If we think tectonic plate movement, fish, lobsters, that kind of stuff, when a little mountain range kind of thing becomes land, they can be cut off, and that reproductive isolation mechanisms will start to evolve and change over time, and they're not going to be able to interbreed. And they've done this with like these weird little shrimp things on both sides of the Panama Canal, basically the Isthmus of Panama. And they're like, hey, these two used to be like the same species because they're on opposite sides of Panama. Let's bring them together. And they're aggressive and towards each other, and they won't mate. And so we say, well, yeah, they're definitely two different species now. So even if you meet again, doesn't mean you're going to make babies. You might be too different at that point in the game. So there are geographic barriers, of course. Think mountain ranges, think oceans, think something along those lines, and that can influence how your species travels and how it reproduces. And this can happen quick or slow. Great Wall of China has changed the plant species on both sides. It's changed all sorts of things because it is a barrier that came up pretty quick. And then tectonic plate movements, of course, moving oceans and moving land would do that as well. So when you have islands that are close to a mainland, like the Florida Keys, little islands off of Florida, you generally don't get new species because they can go back to the mainland pretty easy. But you can get some allotropic speciation. An allotropic speciation can occur if you're a long ways from the mainland because you can't go back to land, like Hawaii, probably formed by underwater volcanoes that made these islands. The individuals, like the birds that are there, were probably carried by the mainland, wind, water, storm, whatever, and those individuals would reproduce and establish populations, but their opportunity to go back and breed with individuals on the mainland is non-existent because the mainland is so very far away, and so you get speciation because they live somewhere no one else does. Oh, here's those two guys I was telling you about. Look at that. Panama, okay, this ocean is now separated from this ocean. This guy lives here, this guy lives here. They look alike, they're the same genus, they're both shrimp, but those two will not mate with each other. That just does not work well. So they're two different species now. Okay, sympatric speciation. Sympatric speciation can occur for populations that are in the same region, so they live together, and they go ahead and become two different species, even though they don't have a physical barrier like an ocean between them. This can occur in a single generation if you get the chromosome number multiplying. Humans, animals, we're not going to tolerate this, but plants certainly can. You have to hope that, of course, some other plant has multiplied its chromosome number, otherwise finding a mate will be really tricky, but, you know, if you're lucky, it can work. This can also occur with no change in chromosome number. They can become two different species, even though they're living in the same area. Okay, next one is parapatric. Parapatric speciation is adjacent populations are becoming two different species, despite being in contact across a common border. And so here you have a border, but I can get around it and still come into contact with somebody else in this population. These can be really big borders. Think like mountain ranges and like lizards that head down two different sides of the mountain range. when we get to the other end of the mountain range, we are so different that we can't interbreed. But we can interbreed if we go back up the mountain range, if that kind of makes sense. It's sort of weird. So hybrids can form in what are called contact zones, which is basically going back up the mountain range. And those are less fit than those on either side of the mountain range, like I was telling you about. So you get these little local pressures on each side of the mountain. The east side of the mountain and the west side of the mountain, they're a little different. And the lizards become a little different with time. And by the time you get to the other end of the mountain, you're too different. So they give the example in your book of these weird, weird little critters called a velvet walking worm, very ugly and kind of cool. And there's two types. There's a giant velvet walking worm, and there's a blind walking worm, and they can interbreed, but their hybrid offspring are sterile. So we can say, yeah, these two guys are definitely two different species. So a lot of that has been microevolution. Can we change the coloration or habits of a lizard over time? Sure, you can, but he's still a lizard, and that's really the kind of the the place where a lot of Christians stand in the evolution discussion, which I'm fine with. But macroevolution is these same evolutionary patterns on a larger scale. We're talking green algae evolving into land plant kind of stuff, really big speciation deals. And there's a few different things that you need to know to kind of understand this concept. And the first evolutionary concept is called stasis. And stasis means not changing. This is the simplest macroevolutionary pattern. It is characterized by little or no change over a really long period of time. Your book gives the kind of sort of funny example of a coelacanth. Coelacanth is a fish. It has like 6 or 7 freaky fins that all have bones like hands, and the thing, they thought it was extinct for like 7 million years or whatever. And then some guys off the southern coast of Africa were like, nah, we've been catching this thing for a long time. It's like 200 feet down in the ocean. It's not extinct. And all the scientists were like, yeah, it is. And the Africans were like, no, it's not. And then they caught one, and all the scientists were like, holy smokes, thing's still alive. So we've got the supposedly, millions and millions, 7 million-year-old fossil or whatever. And then we've got the actual coelacanth that's swimming around being eaten by people in southern Africa. So stasis, we say, well, I guess the coelacanth just didn't change while everything else around it did. So that's stasis. No changing, just stay the same. And then there's exaptation, which is adapting an existing structure for a new purpose. I don't know if we've actually seen this happen. I'm sure we must have. It's got such a neat name, exaptation. The example I think your book gives is like dinosaurs having feathers for insulation because it kept them warm. And then as a neat trick, they could also fly with them. Existing structure, feathers for warmth. New purpose, flight. Awesome. So there you go. Well, there are a few other things that are really important concepts as well for macroevolution. That's adaptive radiation. Certainly Darwin's finch's kind of example of adaptive radiation that's so very famous. And that's one lineage that rapidly diverges into several new species. If you watch my silly little video about my opinion, you've heard this as well. And we see this with honey creepers, which are little birds that live in Hawaii. And each island has different birds, and some eat different things, and so they've eventually become different species. And really, we think it was probably just a few honey creepers that came in from like Asia or something, and just sort of rapidly diversified to fill available niches. And that's exactly what adaptive radiation is. And so then we get the tricky one. This is called a key innovation. This is not a very creative name. A key innovation is a new trait that allows its bearer to exploit a habitat more efficiently or in a new way. Example, the evolution of lungs. It's a key innovation. Everything's got gills or breathe through its skin. And once you get lungs, you can go anywhere on land. And it's amazing. So that's a key innovation. Coevolution is two species evolving together at the same time. That can certainly drive macroevolution. It involves a very close interaction. Think pollinator and plant. Cool. like the shape of the bee must meet the shape of the flower kind of thing. Yeah, length of tongue on moth must meet length of flower. Cool? They have to work together. And so one species acts as the agent of selection for the other species. If the flower becomes longer, then the moth's tongue becomes longer. If the flower becomes shorter, then the moth's tongue becomes shorter. Those two guys are dependent upon each other. So each species adapts to changes in the other species, and the species can become completely interdependent over time. For example, if you took away the bees and the bugs, most, if not all, I can't say all, I don't like the word all, most of our flowering plants would die very soon because they're dependent upon their pollinators. Cool? So that's the end of chapter 17. Points to ponder. How exactly do we get two different species? Uh, when might you expect to see speciation occurring? What is the difference between micro and macroevolution? That's an important question. What are the different types of evidence that support each of these concepts? And that's a chapter 17 for you, so I apologize. I think I ran over by just a little bit, but I do thank you for your time. And we'll talk about this more in class.